The overall goal of this procedure is to select group-specific DNA aptamers to highly hydrophobic small molecules and to use the selected aptamer to develop a sensitive electrochemical biosensor.
This method can help to answer key questions in the aptamer selection field about how to select and characterize group-specific aptamers when targets are highly hydrophobic molecules.
One of the most useful parts of this method is the development of electrochemical biosensors for the detection of targets. These biosensors should also work for the aptamer affinity measurements.
To begin the reaction, add 100 microliters of RNase-free water to the purified aptamer PCR product. Vortex the mixture until the precipitate has dissolved.
Lambda exonuclease reaction is sensitive to the salt concentration, and the PCR product should be purified by ethanol precipitation but not isopropanol.
Place into each of 5 microcentrifuge tubes 5 microliters of the aptamer solution, 11 microliters of RNase-free water, and 2 microliters of 10x reaction buffer. Add 2 microliters of RNase-free water and solutions of 2, 5, 8, and 10 units of lambda exonuclease in RNase-free water to 1 tube each. Mix well with gentle pipetting. Incubate the tubes at 37 degrees Celsius for 35 minutes and at 80 degrees Celsius.